In this module, we will discuss another very interesting and useful technique for the estimation and for differentiating the different DNA molecules or RNA molecules, and this is known as the capillary electrophoresis, abbreviated as CE. Now, this technique of performing electrophoresis in the buffer-filled narrow-bore capillaries, they are normally from 25 to 100 pm in internal diameter. Now you see, the thing is that here we are using capillary electrophoresis rather than using the gel which provides a capillary action there. Here we use the small capillaries which are 25 to 100 nanometer in internal diameter. Now, capillary electrophoresis, it has been used in research since 1980. Many types of separation procedures like single capillary or other, they have been involved. But the recent advances in the technology have allowed the methodology to be more easily adopted to the routine clinical or to the routine laboratory tests. Now, this technique has been now refined to a certain extent. Now, it has got advantage that it is a fast separation of positive sample ID, that is, you can identify the sample positively. It is complete automation is there, and it is also the multi-channel instrument. Now, in this instrument, what happens that you can put different samples and it will identify those samples. It will clearly analyze those samples, completely automated. Now, in the capillary electrophoresis, what happens that the capillaries, they are filled in with a buffer. In narrow bore capillaries, they are filled with buffer rather than the gel. And each capillary is about 25 to 100 micrometer in internal diameter, as clearly as indicated previously. Now, when again a voltage is applied to the solution, the molecules move through the solution toward the electrode of opposite charge, as is the principle of electrophoresis. So the capillaries are used, capillaries are filled with the buffer, and then there the molecules or the sample is introduced into these. Now, basics of capillary electrophoresis is that on the capillary electrophoresis, there is a photoelectrode or photocathode is rather used to measure the absorbency of the molecules as they pass through the solution. So as they pass through the capillary, the molecules, their absorbency is measured by a photocathode, a device which measures the concentration of the molecules. The absorbencies are then analyzed by a computer and they are presented graphically. So we get a graphic result on the computer. Just like shown here, that what happens in the capillary electrophoresis, there are reservoir of the buffer, anode and cathode, and high voltages pass through this. Capillary is dipped in there, and then there are detector, the cathode, and the data which is acquired is, as a matter of fact, submitted to the computer, which gives us a printout of the concentration. This is the capillary electrophoresis apparatus. As you can see, it is completely hooked up with a computer, and then the sample is passed over there. Now, sample application in the capillary electrophoresis is carried out either by high voltage injection. There is a high voltage injection and the sample is filled in, or by the pressure injection. So you can use the both techniques. The high voltage injection can be there, or by the pressure injection, which will be there. Now, comparison of capillary electrophoresis with HPLC, because these are the techniques which are frequently used by molecular biologists, but in the capillary electrophoresis, we consider the power supply is equivalent to an HPLC pump, because in HPLC there is a pump which pressurizes the molecules or the sample to pass through the column. But here what we do, that there is an electric equivalent, or the potential which is supplied, and that is equivalent to HPLC pump, and the instrumentation is completely analogous. Otherwise, it is the same, because it passes through the column and the column measures it. Here, the sample is passing through the capillaries and the cathode is measuring the concentration. This is especially so as the software packages which are used, they control most commercial capillary electrophoresis instruments, and they are based heavily on existing HPLC software. So the software of the both instruments are somewhat compatible. They work on the same principle. Capillary electrophoresis applications are analysis of carbohydrates, analysis of inorganic anion metal ions, DNA profiling, that how the DNA exists there, and the protein identification. So now you can see that capillary electrophoresis is very versatile analytical technique, where the biomolecules, carbohydrates, DNA, and anions, metal anions, DNA profiling, and the protein identification can be carried out. Advantage of capillary electrophoresis is it is a fast technique, and a very small amount of sample is required, and relatively it is inexpensive to run. And the second thing is that it is automated, that you introduce a sample, many samples, and it can analyze them. Disadvantages are that it cannot identify the neutral species. That is the disadvantage. The Joule heating which is there, it cannot do that. And then it cannot discern the shape of the molecule. It cannot give us the shape of the molecule or the molecular structure. So we have seen that capillary electrophoresis, as we compare with HPLC, is very fast, is inexpensive, but has also some disadvantages with it.